This talk is titled Improved Quality of Chlorhexidine Bathing. This is presented by Nasha Talati, MD, who is the Associate Healthcare Epidemiologist for Penn Presbyterian Medical Center, and Pam Tolomeo, MPH, who is the Clinical Research Project Manager for University of Pennsylvania. In this talk, we will review rationale for chlorhexidine gluconate bathing, which will be called CHG for the rest of the talk. We will discuss the concentrations of CHG that we found in a study in our MICU, and talk about methods for improved bathing techniques and practices. Bathing with CHG kills bacteria on patients' skin and reduces the risk of healthcare-associated infections. In particular, patients who are bathed with CHG have a 61% decreased risk of bloodstream infections, including central line-associated bloodstream infections. Better bathing with CHG results in less hospital-acquired infections. In a study that was performed in the summer of 2018 in the Penn Presbyterian MICU, we sampled 33 patients' skin in their neck, axilla, and groin and measured the concentration of CHG. 25% of patients who had had previous CHG baths had no detectable CHG concentration on their skin. And none of the patients in our population had enough CHG on their skin to kill MRSA. This means that while we are performing CHG baths on our patients, we're not getting adequate concentrations on the patients, and as a result, we're not able to kill harmful bacteria that are present on our patient's skin. How can we improve bathing techniques to get higher concentrations of CHG on the skin and kill more bacteria? First thing to do is to make sure you gather all the supplies needed for CHG bathing, including a basin liner. You need 12 washcloths. You need to get the CHG 4% foam and the CHG pump. Also gather warm water in the basin that you will need to rinse the CHG off. Liberally pump CHG foam directly onto the patient's skin. Do not pour CHG into a basin with water, as this will dilute CHG and will make it ineffective. Use enough CHG to clean the patient well. Wash the face and hair in the usual manner. Cleaning the patient is important with a firm massaging motion to make sure that all body sites are covered with CHG. Make sure to start at the jawline. Clean the neck, as this is often the area where patients have central lines. Make sure to clean under the trach ties and around a tracheostomy if a tracheostomy is present. This diagram shows you a systematic way to clean patients. For example, squirt 2 pumps of CHG foam on either side of the neck, 1 on each shoulder and 2 on the chest. Use additional pumps for larger body areas like the back or legs. Depending on patient size, you may need additional pumps of CHG to adequately cover the patients. After applying CHG onto the patient's body, use washcloth 1 to massage the CHG into the neck, the shoulders, and the chest area. Then use washcloth 2 to clean the front and back of each arm and the axilla. After squirting CHG onto the groin, abdomen, and inner thigh area, use washcloth 3 to clean this area and also clean the proximal 6 inches of the Foley catheter if a Foley catheter is present. The groin has a lot of bacteria and can be contaminated by bowel movements. Therefore, it is critical to clean this area well. Use cloth 4 for the front and back of one leg and cloth 5 for the other leg. After the CHG has been thoroughly applied on the skin, take a warm, wet, clean washcloth and rinse off the CHG, starting with area 1 and moving through till area 5. Be sure to discard each washcloth in the soiled linen bin after use. Allow CHG to dry for 2 minutes before rinsing each area with a washcloth dipped in warm water. Gently pat dry. Do not apply soaps, lotions, powders, or deodorant other than those stocked in the clean utility room. These will inactivate CHG. Documentation of CHG bathing should be performed in pen chart. We are providing a huddle sheet that has key information and ways to contact us and contains the policy and link to the REDCap video.